All right, guys, let's talk about the mitochondria. Say mitochondria. And really, what we need to know today is the mitochondrial structure and how we can make ATP. Because remember, the mitochondria, not that I enjoy it, but he is the powerhouse. And the reason that the mitochondria is referred to that powerhouse is because he is going to make the energy for my cell. And then, of course, we all know the energy for my cell, this is going to be ATP. And so what we need to do today is talk about ATP and structure, and then, of course, like I said, the mitochondrial structure. So let's talk about ATP first. All right, ATP. ATP stands for adenosine triphosphate. Okay, now adenosine triphosphate. So let's kind of break through this. Here's my adenosine, and then triphosphate. That just means there's going to be 3 phosphate groups. So let's start out with my phosphate groups. We know phosphates, PO4. So here's one phosphate. And then let's draw our next phosphate. And then our third phosphate. Now, if you guys remember, my phosphates, okay, they have a bunch of negative charges, okay? So this is my phosphates. And my phosphates, by characteristic, remember, were very unstable. And the reason they're unstable is because of all of these negative charges. Remember, oxygen is super electronegative. And so if you can imagine putting 3 magnets next to each other. So like, let's see, here's a magnet with a negative charge, and here's a magnet with a negative charge. Since they're both negatives, that means they repel each other, and they're going to try to get away. Well, now you're looking at 3 magnets with negative charges. And since they all have negative charges, and they're very, very negative, that means they're going to want to break off from each other really, really fast. And that's how we're going to get our energy, because you guys know when we break a chemical bond, that releases energy for my cell. All right, so let's keep going. So there was my 3 phosphates. Then I have this guy right here. This guy, this is my ribose. And we know ribose is a sugar. And the reason we have a sugar right here is because it's sticky. And that means we can use that sugar to stick my phosphates to my adenine group. And my adenine group is going to look something like this with some nitrogens here. And then we have another nitrogen right here. So that's NH. So this is going to be my adenine. And my adenine, if you recall, he is a nitrogen base. And for fun, he is a purine because it has 2 rings. And so now that I'm looking at this ATP molecule, I can really kind of draw it like this. So here's my 3 phosphates. And then here's my adenine group. And remember, all of these phosphates are super negative. And since they're super negative, they don't want to be around each other, right? They want to repel each other, like these magnets over here and over here. And so the phosphate group that's the easiest one to break off is this guy, because everything else is attached to this adenine group. And so since he's the one that's kind of the least attached to everything, then he's going to break off. And then as soon as I break him off, I'm going to get energy out of it, which is how I get the energy out of ATP. So if I draw a really simple diagram, so here's my ATP. And then what's going to happen is I'm going to break off that phosphate. So now I have a phosphate floating out here. And now I only have 2 phosphates. This is now ADP, or adenosine diphosphate. because there's only two. But because we're not a wasteful people, I can take that ADP, I can add a P to it, and I can get back to ATP. And that's how I'm going to recycle my energy really fast. But take home message, because these phosphates are so negative, they want to be repelled, and so this guy is the easiest one to break off. Could I break off that second one? Sure, I could, but he's a little bit more stuck, and so he's going to stay attached a little bit better. And then this guy over here, this last one, he's really, really hard to break off because he's not being repelled by anything. So it's this last phosphate that we break off all the time, because that will give me the most energy out of everything. Now, how do we make ATP? Well, ATP, guess what, is facilitated by a chemical reaction. So I'm going to do the hydrolysis. Oh yeah, let's do that. So the hydrolysis of ATP, because I want to break off that phosphate. So here's my ATP, and then we know hydrolysis, hydro is water, lysis is a break. So I'm going to use water, and then I'll get ADP plus P plus my energy. And so that's going to be the chemical reaction for how I get the energy out of ATP, is the hydrolysis of them. So now what I need to do is I want to know, how do I make ATP? All right, well, how I make ATP, I get to use an enzyme. And so the enzyme that I'm going to use is called ATP synthase, which makes sense because ATP is the molecule I'm using. Synth means to make, and ACE is referring to an enzyme. And we know that we read science words backwards, and so we're going to use an enzyme to make my ATP. And what this enzyme looks like, he's actually pretty cool looking. He looks kind of like this. So he has a big box, and then he has this little guy, and then this part. So this right here, this is my ATP synthase. And remember, whenever we used to draw enzymes, we'd always draw it kind of like this. And we know this was my enzyme, and then there was my active site right here, and then this is where the substrate would fit in. And so what I'm going to do now is I'm just going to do the reverse. I'm going to take my ADP plus a P, and that will give me ATP. All right, well, if this is my enzyme, that means I need to have an active site. And so really that active site is right over here in this corner. There you go, we'll make it a little bit different. There's my active site, right? So this is where my substrates are going to come fit in. So these right here, my ADP and my P, these are now going to be my substrates. And so I can have one, this could maybe be my ADP, and then I can have this guy right here, he can be my phosphate. And so what's going to happen is this ADP and this phosphate, we'll kind of draw a picture, this ADP and this phosphate are going to come sit inside my active sites. So there's my active sites, and so now the ADP and the P are going to go sit in there. Remember, this is called my enzyme substrate complex. And so now that I've had my enzyme substrate complex, this thing, here's my ATP synthase. Whoops, let's try that one again. There's my active sites. And so then now that the reaction is complete, I get my ATP. And that's just how that enzyme works. Now, how it works, though, is we're going to have this top part up here. He actually gets to spin. And so when he spins, that's going to force these two substrates together. So that way I get this ATP in the end. And that's how ATP synthase works, is once the substrates fit inside, then this top part spins. When he spins, that forces them together, and then I get my ATP, which is my product. Okay, so now what we're going to do is we're going to talk about my mitochondrial structure. All right, so let's talk about the mitochondria. Mitochondria. So mitochondria structure. Mitochondrial structure. All right, let's draw this cute little guy. So remember, he is a big dude, looks like that, kind of like a bean. And then I have that squiggly stuff in the middle. All right, mitochondrial structure. First, this is my outer membrane. This space right here, this is called my inner membrane space. This right here, what we should be familiar with, is called my cristae. And then this part right here, the middle of it, this is called my matrix. And so where this ATP synthase sits, it actually sits inside my cristae. So we could kind of talk about that. So here's my cristae structure. Now my cristae structure is actually a bilayer, which we've seen before. So I have these cute little phospholipids. And then sitting inside my phospholipids is my ATP synthase. And I'll make my active site over here. There you go. And then there's my ATP synthase. And so what happens is, here are my substrates. Oops. Here are my substrates. My ADP, that's a triangle. And here's my P, here's my triangle. And so what they're going to do is they're going to go sit inside this active site. So let's draw. Here's my phospholipid bilayer. Here's my ATP synthase. And so now those two substrates are going to go fit inside the active site. And once they fit inside that active site, this top part of ATP synthase starts to spin. When he spins, then that is going to force those two substrates together. And when they get forced together, now I get to make my product, which is ATP. And that's going to happen at this cristae. So right here, if this is my cristae, this part, this is my inner membrane space. And then down here, this is going to be my matrix. And so if I were looking at this picture, like let's say right here, here's the top part of the ATP synthase, and then there's the bottom part. Now the fun thing about the cristae is that that's going to have a lot of ATP synthases, so I can make a lot of energy. But in all reality, that's how ATP synthesis is done. It's made in the mitochondria. ATP releases a lot of energy because of all of these negative oxygens. And since this last one right here, that'd be the easiest one to break off, that's where I get the most energy. So I get the most, go ahead and write it, most energy by breaking that third phosphate off, because he's going to be the easiest one right there. The other two, I'd have to use too much energy to break them to get anything out of it. And so the chemical reaction of that is the hydrolysis of ATP. So I have ATP in water, that'll give me ADP, and then the energy that I want. And how I'm going to make that ATP in the first place is by ATP synthase. And ATP synthase is an enzyme, so it works just like normal. I have my two substrates right here. They go fit inside that active site. Once they fit inside that active site, that top part of synthase spins, forces them together, and then I get ATP as a product. And where that's going to happen in the mitochondria is there's my mitochondrial structure, my outer membrane, my inner membrane, the cristae, which is really, really important, and then the matrix. And so the cristae structure, there's my inner membrane space, and then there's my matrix. So there's my ATP synthase right here. The substrates, the ADP and the P, go sit inside the active site. Once they sit in there, that thing starts to spin, forces them together, and then ATP is released as a product. All right, I hope that helps.